Hello, and welcome to the Binfie Conference 2020, Session 2. This is a web conference organized by Nabil Ali Khan, Andrew Page, and myself, Lee Katz. I want to give a lot of credit to Nabil for setting up most of the structure of the conference, including the Discord space, the website, the registration, and many, many other things. So thank you very much. For this week's session, I want to give credit to Andrew also for getting us started on a special bioinformatics trivia night during the social hour in Discord. And I have to give you the fine print that this is a conference not sponsored by my employer, CDC, or by the Quadram Institute. And the opinions expressed here do not necessarily reflect those by CDC or Quadram. We are pleased to have you all here today. I should mention that the Zoom session will be recorded and the video will be made public. And so if you do not want to be videoed, please close your video now. Also, please remember to keep your audio muted. We hope to go beyond the standard webinar. We have a lot of social mixing, and just to that end, I think it's a critical part of the meetings when we're in actual personal space, and I hope that we can translate that here online. That interaction will be driven by the Discord server running alongside these Zoom meetings. So please sign into that as well. The link is in the group chat of the Zoom session and on the website. Please move to the Discord after the Zoom meeting, where we will have a virtual coffee break where we can mix and ask more in-depth questions of the speakers, and we can talk some more science. Or just drink virtual beer or coffee with me. So as I mentioned earlier, new for this week, we'll see if we can keep it going, but at least for this week, we're gonna host a Binfie trivia hour, and we'll give more details on that after the speakers. Today's session is going to be tied together by genomic epidemiology. We're going to have some very exciting talks from Melinda Ashcroft, Taylor Paisley, and Federico Palma. And so, Melinda, do you want to kick us off? Oh, I'm sorry. I have an introduction here for you too. So for those of you who don't know Melinda, Dr. Melinda Ashcroft was awarded her PhD from the University of Queensland, Australia in early 2019, investigating the roles of mobile genetic elements, antimicrobial resistance, and DNA methylation in the evolution of Escherichia coli lineages. Melinda is now a postdoc research fellow in microbial bioinformatics at the Peter Doherty Institute for Infection and Immunity at the University of Melbourne, Australia. Her work focuses on genomic epidemiology, transmission, evolution, and antimicrobial resistance of pathogenic Neisseria species. Melinda is also passionate about science communication and is a current Australian Society of Microbiology Communications ambassador. And guys, Melinda is talking to us in the middle of the night at 2 a.m., I believe. So please be very kind to her. And one more really cool thing about what we're doing is you can click an icon to applaud her too at the bottom of your Zoom window. So please give a round of applause to Melinda with your Zoom icons, and please take it.
All right. So thank you very much for the introduction, and I hope everyone can hear me okay. Excellent. And so today I'm going to be talking about some of the most recent work that I've been doing, looking at how whole genome sequencing can reveal, or did reveal, limited within host variability in Neisseria gonorrhoeae from multiple anatomical sites. So just a really brief background on Neisseria gonorrhoeae. It's an urgent public health threat, and it's the causative agent of gonorrhea, which is the second most commonly sexually transmitted infection worldwide, with more than 100 million cases globally each year. So untreated infections can lead to severe sequelae, particularly in women, and can result in pelvic inflammatory disease, ectopic pregnancy, and infertility. In addition to this, it's also the leading cause of, one of the leading causes of neonatal conjunctivitis, which can lead to blindness. And in all sexes, it increases the risk of getting and transmitting HIV. As well as this, the incidence in Australia and worldwide has been rapidly increasing over the last decade, which we can see here on the left. And between 2014 and 2018 in Australia alone, the incidence almost doubled. As well as increasing incidence though, we're also seeing increasing resistance to our first-line empirical treatments, which in Australia are ceftriaxone and azithromycin, resulting in this superbug status, where in 2019, the Centre for Disease Control and Prevention labelled them as an urgent priority pathogen. So mathematical modelling and genomics provide a really powerful method for investigating transmission dynamics of Neisseria gonorrhoeae. However, this comes with some caveats or assumptions. So most of these studies don't consider that there can be multiple anatomical sites, multiple infections at different anatomical sites, or they assume that patients with infections at multiple anatomical sites are actually infected by the same strain. And that this therefore leads to a potential underestimation of transmission dynamics, which is an issue because these studies actually guide public health interventions. So our aims were threefold. Firstly, we wanted to investigate the within-host genetic diversity across multiple anatomical sites. And to do this, we had 37 individuals and we had 2 to 3 isolates per individual from a range of sites, including the oropharynx, urogenital and anorectal regions. And these isolates were obtained between 2011 and 2019. Secondly, we wanted to investigate the genetic diversity of multiple colonies within individual samples. And lastly, we wanted to propose a minimum methods framework for future genomic studies that investigate genetic diversity or transmission. But how do we actually define within host genetic diversity? And so to do this, we created a core genome SNP alignment using SNPy with default parameters. But what does that actually mean? So this image is courtesy of Torsten Seaman. And we have our reference genome along the top here in blue. And then we've mapped the reads from each isolate to this reference genome and called the core genome, i.e. the positions that are conserved in all the strains, which we can see here along this line. We've then called the core genome SNPs, which are mutations or differences in at least one isolate within this core genome. And then lastly, we're looking at unambiguous core genome SNPs. So we're removing things such as ends or degenerate bases. And this leads to a core genome SNP alignment, which we can see here. We've got our SNP alignment is 4 SNPs. And we can see between isolates 1 and 2, they differ between 2 SNPs at position 2 and position 4, or 4 SNPs between isolates 2 and 3, and again 4 SNPs between isolates 3 and 4, and 1 SNP between isolates 1 and 4. So one of the biggest factors affecting the core genome SNP alignment is the choice of reference genome. And this is because as the differences between a reference genome and an isolate increase, fewer reads map against the reference genome, and it has the potential for missing informative SNPs, such as those that can define clades or lineages. And it can also result in the overestimation of pairwise SNP distances between isolates. However, this is really limited by the availability of complete reference genomes in public databases. So for example, there's only 33 complete Neisseria gonorrhoeae genomes. However, for example, there's more than 1,000 E. coli genomes. So in some studies, it may actually be prudent to generate your own complete genome from one of the isolates within your study using, for example, long-read sequencing. One of the other major issues when using complete reference genomes is that they're often missing higher level annotations such as repeat regions and mobile genetic elements, and this therefore requires manual annotation. So for example, if we wanted to annotate phages, we could use a tool such as FASTA, and genomic islands such as, we could use a tool such as Island Viewer 4. In this study, we actually identified putative repeat regions by aligning the reference genome to itself using the mask auto function in SNPy, which generates a bed file of coordinates to mask. So in this study, we actually looked at 2 different genomes, or 2 different reference genomes. Firstly, we used the internationally recognized antimicrobial susceptible complete genome, FA1090, which was sequenced in the early 2000s, and it's used extensively in Neisseria gonorrhoeae genomic studies. And then we used an Australian complete genome, this AusMDU genome, which was isolated in Victoria in 2017. And we predicted that this might actually be a closer reference genome. So another major factor that affects the core genome SNP alignment is SNPs within repetitive regions and those within mobile genetic elements. So when we plotted the density of SNPs within the core genome alignment against the FA1090 genome, we actually found several high density SNP clusters. So here on our y-axis is the number of SNPs, and on the x-axis is the position of these SNPs in the FA1090 genome. And after filtering of SNPs in these regions, which we had to manually annotate firstly for both of the complete genomes, we can actually see that several of the high density SNP clusters had dropped in size. So particularly these ones here and these ones here. Additionally, we compared what happened to the maximum pairwise SNP distances of isolates that we expected to be the same strain, given that they were from the same individual. So initially, the maximum distance of isolates from the same strain were 80 SNPs. However, after filtering, this reduced to 40 SNPs. But this is still larger than most estimates from previous Neisseria gonorrhoeae studies, which have a range of between 3 to 22 SNPs being used to define isolates as the same strain. So again, we examined the density of SNPs within the core genome SNP alignment, but this time against our AusMDU reference genome, which had several high density SNP clusters also. And again, after filtering of SNPs within repeats and mobile genetic elements, we can see a decrease in these high density SNP clusters, particularly these ones here and these two down here. And again, when we compared the maximum pairwise SNP distances of isolates that we expect to be the same strain, the maximum distance was 64 SNPs before filtering, and after filtering of SNPs in repeats and mobile elements, the distance was 36 SNPs. But these maximum pairwise SNP distances still seemed really large, given isolates within each isolate were supposed to be the same strain. So we also investigated the effect of recombination on the core genome SNP alignment. And here we found that filtering of SNPs in recombinant regions removed between 25 to 31% of the core genome SNP alignment, depending on the reference genome. However, this still left more than 11,000 SNPs to build the phylogeny. And in fact, after filtering the core genome SNP alignments, they differed in size by only 300 SNPs. So additionally, the maximum pairwise SNP distances of isolates that we assume are the same strain were now between 18 to 20 SNPs. So given the minimal differences between choice of reference genome for this specific data set, we continued using the AusMDU reference for the rest of the project. So the final factor that we wanted to investigate was whether isolates from other individuals were actually affecting pairwise SNP distances. So here we used an individual specific reference genome approach where for each individual, one isolate was chosen as the reference and de novo assembled, and then the sequence reads from other isolates within that individual were mapped to the reference and SNPs were called. So this then resulted in 37 small core genome SNP alignments instead of one large core genome SNP alignment. However, care needs to be taken when using this approach, which I've got shown here, as the reference genome is a fragmented assembly, and so repeats and mobile elements are very difficult to identify and thus weren't filtered from the SNP alignment. So what were the actual results of this part of the study? So here we've got a maximum likelihood phylogenetic tree of all 105 isolates generated from the core genome SNP alignment with multi-locus sequence type and multi-antigen sequence type plotted alongside. And what we found was that in 35 out of 37 of our individuals, or 95%, isolates from that individual clustered together, and they had identical multi-locus sequence type and multi-antigen sequence type profiles. However, we had 2 individuals that had multiple co-infection strains. So individual 17, which is here in teal, has a urogenital isolate that is in a different part of the tree to the oropharyngeal and the anorectal isolate. And similarly, for individual 29, which we see down here, we have our urogenital and anorectal isolates clustering together, but in a different part of the tree entirely is the oropharyngeal isolate with a different MLST and multi-antigen sequence type. The last thing that we noticed was that multiple individuals actually clustered together in the tree, and that these individuals are likely to be part of the same sexual network, which is suggestive of putative transmission clusters. So in our first plot here, we show that the maximum core genome pairwise SNP distance to define isolates as the same strain was 18 SNPs. And we can see that our two individuals with multiple co-infection strains had pairwise SNP distances that greatly exceed this 18 SNP threshold. In our second plot, we show that the maximum individual specific pairwise SNP distance to define isolates as the same strain was 13 SNPs. You can see this here. And again, we see that our two individuals with multiple co-infecting strains had pairwise SNP distances that greatly exceed 13 SNPs. Sorry, down here. Lastly, we wanted to investigate the genetic diversity of isolates between hosts. So while we see a large spread of pairwise SNP distances, we did identify a number of isolates with maximum pairwise SNP distances around 0, indicating the likelihood that these isolates are similar, which we investigated in more detail. And here we found that isolates from several patients clustered together and were almost indistinguishable. They had highly similar multi-locus sequence type, multi-antigen sequence type, and antimicrobial resistance sequence type profiles. And in fact, we had 4 distinct clusters. So in cluster 1, all isolates had the same sequence type profiles, except for those within patient 9, which had a different multi-antigen sequence type. However, the maximum within cluster pairwise SNP distance was 18 SNPs. In cluster 2, we see something very similar, where we have almost identical sequence types for from 4 isolates within 3 individuals. And we have a maximum within cluster pairwise SNP distance of 11 SNPs. Again, for cluster 3, we again show very similar sequence types for these isolates, except for a couple of differences with the multi-antigen sequence type and a pairwise SNP distance of 13 SNPs. And lastly, in our final cluster, we actually see differences both in the multi-antigen sequence type and antimicrobial sequence type, but we have a maximum pairwise SNP distance of 15 SNPs. And so what this is saying is that we're showing the likelihood that individuals within each cluster are involved in the same sexual network, and that the maximum pairwise SNP distance for each cluster is in the same range as the within host diversity. So what this means is that the within cluster and within host genetic diversity is so similar that we can't actually determine based on genomics alone whether infections at each anatomical site are the same infecting strain or whether they represent reinfections from the same sexual network. So our second aim was to investigate whether multiple colony picks from the same sample were the same strain. And to do this, we chose 3 to 4 colonies from 4 to 5 primary oropharyngeal, urogenital, and anorectal cultures. So here is our maximum likelihood phylogenetic tree generated from the core genome SNP alignment of all of these isolates against our OzMDU reference genome. And what we found was that there was a maximum pairwise SNP distance to define isolates as the same strain of 2 SNPs, and that all isolates within each sample clustered together. Notably, we had 1 individual that had both oropharyngeal and anorectal samples, and that isolates from both of those clustered together. And we had 4 individuals that had isolates that clustered together, again, similarly, likely suggesting that they're from the same sexual network. So again, we investigated whether isolates from other samples were affecting pairwise SNP distances. And so instead of 1 large core genome SNP alignment, we generated 14 smaller core genome SNP alignments, 1 from each sample, which we show here. And so here we see that the within sample diversity is actually really similar to the within host diversity that we previously characterized. And we show that the maximum within sample pairwise SNP distance to define isolates as the same strain was 12 SNPs. However, we also identified 3 samples that had high density SNP clusters, and that these were actually associated with phages or recombination. And we actually had to manually exclude these regions from the SNP alignment, as for example, with this urogenital sample 2, the SNP distance was actually 29 SNPs originally. So our final aim was to propose a minimum bioinformatics framework for future Neisseria gonorrhoeae genomic studies. So in part 1, we first proposed the use of a close reference genome, which is either publicly available or is generated as part of your study. Next, we generate a core genome SNP alignment. And while there are many tools that can perform this, it's important to provide full details of the tools, the versions, and the parameters used. Thirdly, SNPs within repetitive regions and mobile elements should always be filtered from the SNP alignment. However, this usually requires manual annotation of these regions in the complete genome. And lastly, SNPs within recombinant regions should be filtered from the alignment, provided that it does not remove too much of the phylogenetic signal. And then in part 2, if investigating within host or within sample isolates, we propose that multiple smaller core genome SNP alignments are generated per individual, and that these alignments are manually examined to exclude SNPs within high density SNP clusters. So ultimately, pairwise SNP distances differ across studies due to the size of the core genome, the closeness of the reference genome to isolates within the data set, filtering of SNPs in repeats and mobile genetic elements or in recombinant regions, and the use of outbreak or epidemiologically linked isolates. And that we really need to provide full methods and parameters to ensure reproducibility and allow comparisons between studies. So just to conclude this talk, in 95% of the time, we found that isolates within an individual were the same strain, and that the within host diversity was a maximum of 18 SNPs difference at the core genome SNP alignment, or 13 SNPs different in the individual specific SNP alignment. We found 4 clusters of isolates from individuals that had maximum pairwise SNP distances that were comparable to the within host variation, and therefore these were part of the same sexual network or transmission cluster. And we found that the within sample genetic diversity was comparable to the within host diversity, where each colony pick was indeed the same strain, and that it was a maximum of 2 SNPs different in the core genome SNP alignment, or 12 SNPs different in the sample specific SNP alignment. So this really says that our study actually agrees with previous modeling studies that assume isolates from multiple anatomical sites within an individual are actually the same strain. And so lastly, analysis of whole genome sequencing data alone can really provide uncertainty into whether isolates at different anatomical sites within an individual are actually the same infecting strain, or whether they represent reinfections from the same transmission network. So just to conclude my talk, I just want to acknowledge the Williamson Research Group and my supervisor, Professor Deborah Williamson, as well as Darren and George, the Department of Health and Health Services Victoria, our collaborators at the Melbourne Sexual Health Clinic, especially Associate Professor Eric Chow and Professor Christopher Fairley, and the Microbiological Diagnostic Unit Public Health Laboratory. Thank you.
Thank you so much, Melinda. That was a really good talk. You have several questions, so I'm going to try to read them in order, and thank you for Nabil for compiling them.
Excellent.
When reducing the alignment from SNPs to unambiguous SNPs, did you cut out the whole column that included an N, or did you exclude them pairwise?
The whole column. Yeah, so if there's even one N or one degenerate base, the entire column is removed.
That makes sense for core. And then another question is, did you hypothesize that these different isolates would infect different sites? If so, what would be the clinical or biological implication of that?
We didn't, but there has been some work on looking at whether isolates at different anatomical sites are actually the same infecting strain or are strains, from their sexual network. That's not something that I've looked at specifically in this study, but it is definitely something that Professor Christopher Fairley and Associate Professor Eric Chow at the Melbourne Sexual Health Centre have been quite involved with.
Okay, great. How does the pan-genome look between these within sample and GONO?
That's a really good question. So I've been quite lucky in that in all of these isolates, the pan-genome looks almost identical. I didn't notice any isolates within an individual having picked up a plasmid or dropping a plasmid or anything like that. But that is definitely something that you should look for when you're doing these sort of studies, and definitely take that into account.
Okay, I'll give you a few more, and I realize it's in the middle of the night. Thank you so much.
That's all right, it's fine.
For the within sample diversity, do you have culture bias or anything that might reduce the subpopulations?
I don't know, actually. That's a really good question. I don't know. All of the samples are prepared the same way, and they're all done at the Melbourne Sexual Health Centre and then sent to us. So there definitely, I guess there could be bias, but I guess, we would hope to see something like that if we would, with multiple colony picks. So yeah, that's a really good question, and I don't know.
Yeah, these are really tough questions. So
definitely something that I should think about though, and maybe look into. So thanks for that.
Okay. How do you filter the recombinations from the alignment, and how do you define them in the first place?
So we used, in this specific instance, we used gubbins to identify and remove the recombination. But there are other tools available, for example, GATK, that do similar things. In this case, we just use default settings for gubbins. But I have seen other people who prefer, for example, GATK and use, for example, a cutoff where if there's more than, say, 3 SNPs within a 100 base pair region, then all of those SNPs are discarded. So yeah, I guess it really depends on the tool that you're using, but in this case, we just use gubbins with default settings, and it actually provides the alignment after filtering.
Okay. Can we do one more? Which tool did you? Oh, sorry. So you just told us to use gubbins, right?
Yeah.
So is there any indication that the mobile elements might be associated with adaptation to a particular site or to particular sites?
Perhaps. So the oropharynx region in particular has a lot of commensal Neisseria strains, and Neisseria species in general are exceedingly good at exceedingly good at recombination. And so, they definitely could pick up mobile genetic elements that confer, for example, resistance. And that's something that I guess has been touched on in the literature in the past, and is definitely something that we could look at in the future.
All right, thanks so much. I think you got a huge project of questions. I think you have a lot of good answers. There are a lot more questions, actually. Hopefully it doesn't keep you up too late tonight reading through them, but I'm sure they'll be there waiting for you in the morning if you want.
No. I'll get onto it now.
That's awesome. And everyone, please hit your applause buttons. And thank you, Melinda.
Thank you very much.